it's a promising, strategy. It remains to be seen how effective it is going to be. So they just got to keep trying it on patients until we actually have a vaccine that would allow people to generate those antibodies. Okay. So, Jalen, Josh, Gideon, Ahmad went. Who else? Are we missing anybody? Did anybody not get
I
to tell us your idea?
think we all went.
Okay, everybody went. Okay. All right. Well, look, as a group, okay, given those ideas, which ones? So we're not going to talk about the coalescent serum part because everyone's already doing that. Like that's already happening. So if you were a researcher and you're coming up with ideas like we're doing right now, you would say, okay, what are my ideas? And then are there any people out there doing it? Right? And if there are already people out there doing it, then unless you found a way better way of doing it, probably better to try to think of another idea. So let's put our thinking caps on. Let's just talk here as a group. Any other ideas that you would have? Like what would be your second option, each one of you? And we can kind of go in order. Go, Alex.
So
Whoa.
we know it reacts with the ACE2 protein. Could there be like any other protein that could be involved within like the chain sequence of the virus affecting the cell? Or is it just that one?
What do you mean? Explain.
Because it enters through the ACE2 protein, right?
Mm-hmm.
Could it be like I don't know, maybe there's like specific ribosomes that create the virus, maybe we could interact with those, or we can like get down more to the specifics of how the virus is being made in the cell and try to stop it from there. Or I guess we could go the other way and be more broad.
So kind of like chemo with cancer in a sense.
Mm-hmm.
Yeah,
I
but
was kind of, yeah.
but there's always the fallback of like we could do let's say collateral damage,
Mm-hmm.
and then but basically what the virus is doing where people's immune systems are overreacting. Actually, could we actually find a way to suppress, like immunosuppress? What's it called?
That's a great idea.
To
So
suppress the immune system.
how do you suppress
Intro.
the immune system in the patients that seem to be the most at risk? I think that is a brilliant idea because really what's happening when you think about how the virus is killing you, right? It's when it gets into your lung tissue, your lungs, and it really starts to infect the cells. They're called lung epithelial cells. It not only kills the cells, but it causes the cells to release these proteins called cytokines. Okay, you guys know what cytokines are?
No.
Somebody look up on one of you guys look up on Google what a cytokine is. CYT
Isn't it like
O
the
K I
suicide?
N E S.
Cytokines.
Yeah,
Cytokines are a broad and loose category of small proteins important in cell signaling.
right. So, all right, now add to that Google search cytokine storm, 2 words.
Cytokine what?
Storm, like a storm outside.
Cytokine storm. Cytokine.
Overproduction of immune cells.
Yep.
Right. So this is as I've been talking to my medical colleagues who are actually treating patients who are coming into hospitals, let's say in New York and other places. One of the things that they see in patients that do really bad is this cytokine storm, right? That means that in their lungs, they get this release of all of these proteins. And what do cytokines do? Read a little more about them.
Okay. Interact with cells of the immune system in order to regulate the body's response to disease and infection.
That's
So
right.
kind of like white blood cells.
Right. So white blood cells make them and respond to them, and all cells can kind of send them out. There's basically like the signals to let your immune system know like something's wrong, right? So when you get sick, when you get a cold, right, you get a fever, right? And the reason you get a fever is because
Your
your
body's
cells
trying to
literally
kill off the virus.
they start heating up, and producing these things called inflammatory cytokines. They're what makes your cells literally get hot and bothered, right? And they will cause your cells to replicate. So it causes certain types of cells, particularly your immune cells, like your T cells and your B cells, to make more of themselves. So you have all of these different molecules. They have different names, like interleukin 2 or interferon gamma, right? Things like that. They have different names that researchers have given them over the years as they've discovered them. But they're really important because the way your cells communicate in your body is through these signals, these proteins that get released into your bloodstream, right? So when you have an infection and you feel your lymph nodes are swelling, or let's say you got these 2 lymph nodes here that swell, what will happen is they're swelling because you're getting more T cells and B cells growing in those lymph nodes. And those cells are going to be being trained to seek and destroy the virus infected cells, right? And when you do that, though, when you release these cytokines, it can have some bad effects. If your fever gets too high, if you have too much of it happening, it can cause like a shock type of syndrome and shut down your organs. And that's what seems to be happening in the lungs of some of these patients. They get a massive cytokine storm that creates this like shock syndrome in their organs, so that they can't even function anymore. They just break down. So whenever you lose the function of an organ, it becomes very life-threatening, because even if you're on a ventilator, if the cells in your lung aren't functioning, you're not going to get any oxygen across the barrier there, and you won't get oxygen to your tissues, and you'll die. Your organs will just start shutting down without oxygen and energy. And that's what's happening. So if we could find a way to somehow monitor or like down regulate these cytokines, prevent the cytokine storm, I think that would be a good idea. Okay. Any other thoughts? Any other ideas?
Can we possibly treat the coronavirus like the same way as we can treat like a critical asthmatic person?
Like with steroids, right?
Mm-hmm.
That's interesting. That's a good question. I mean, you know, the steroids are actually there to relax and open up your airways. So in this case, I think it would be bad to actually open up the airways when you have a virus infection, right? Because you didn't give that virus like more room to operate, right? If you and I think that's part of the challenging nature of folks who are asthmatic, who seem to be at higher risk, because you already are dealing with decreased lung function, right? You've got sort of these closing of the lung airways, and so that constriction of the bronchioles is bad. So you don't have a lot of lung to give up, right? And so if the virus is already affecting your lung, you already maybe have lost 10%, 20% lung function with the asthma. And then if you lose another 50, 60% with the virus, you're gonna have real trouble breathing. So I think giving people steroids would actually probably be a bad thing, because you would be opening up those bronchioles and then causing potentially more infection. Any other thoughts? Okay. Well, look, let me share with you guys my idea. Do you want me to share it? Can I share mine?
Yes, please.
Okay, cool. So look, I've been thinking about this. I'm a molecular biologist, been thinking about this a lot. And one of the things I agree with, Alex mentioned the ACE2 protein, right? And the spike protein. So I'm going to share my screen, okay? And I'm going to show you how my idea evolved, okay? Can you guys see my screen?
Yes.
Yes.
Okay. So here's actually a paper that hasn't even come out yet. It's actually, let's see, this is in press, right? This article is in press. So that means it's still being reviewed. It's going to come out on next week. But I got a preview copy of it because I wanted to learn the difference between SARS coronavirus and COVID-19 at the surface. Because my hypothesis was, if you can block, like you said, with an antibody or with a drug, right? The interaction between the SARS spike protein and the ACE2 protein on the surface of cells, then you might actually be able to stop the virus. Now, this a unique nature. Maybe one of you guys mute. Hang on, maybe mute for a second. A unique nature of this coronavirus appears that it uses another receptor. It's called TMPRSS2. It's another gene other than ACE2 that helps get it in. Now, this paper talks about the neutralization from coalescent serum, right? So people have already been doing that. But one of my ideas is, can I make something that looks like SARS on the outside, but isn't SARS on the inside? So it's no danger of being infected. So it's like a particle that looks like a virus that I can put the SARS spike protein, and this other protein is called the E protein or envelope protein, on the surface, and see if I can make an what's called a screening assay where I can test drugs to see that block this interaction. Because actually my idea is you're going to have people with different levels of antibodies, and the virus can really mutate well against antibodies. But if you find a part on the virus, on the spike protein, that's very conserved and very consistent, then maybe you can make a drug that will stick to it and prevent it from sticking to ACE2, right? Kind of like a drug that works like an antibody, like that will work like a vaccine. Okay, so in reading this paper, you find that there is actually a drug out there that this protein ACE2 is a protease, and TMPRSS2 is also a protease. These are actually molecules that are on the surface of the cells that are actually proteases, right? So what this group did that I thought was really interesting is they started looking at SARS, the old SARS and the new SARS, and they looked at it on it, and then they also looked at another unrelated envelope from another virus called Vestibular Stomatitis virus. It's called VSV. It's just another family of viruses not related to SARS, but we commonly use it in research because the envelope of VSV can infect like a lot of different cells, right? So what they did was they took what's called a lentivirus, like we're working with, HIV, right? And they made the viral particle, okay? In this case, the kind of inside of it is lentivirus. It's actually HIV-like, right? But they put on the surface, it's called pseudo typing. That's what we call pseudo typing. That's a word that we mean. That means when you take the envelope from one virus and put it on the surface of another virus. That's called pseudo typing. And we've been doing that for like the last 20 years with HIV. So for example, when we made the lentiviral vectors, in order to make them not really infectious like HIV was, we changed out the envelope. So HIV has its own envelope protein called GP120, right? Now, if we just left GP120 on the HIV particle, then it could potentially cause immunodeficiency. It could kill you. But if you took off the envelope and replaced it with this vestibular stomatitis virus envelope, VSVG, not only will you make it not kill you, you make it infect a lot of different other cells, which could be actually useful for medical purposes. Because just like we're trying to make a lentiviral vector that can transform the gene expression in tumor cells in our main project, this you could actually make a vector that can infect a lot of different cells, right? So what they did was, just like you were able to change the HIV envelope out, the GP120 out for VSVG, they changed it out for SARS spike protein, and then now COVID-19 spike protein. And you can see the different ability of that virus to infect different cells. And so what they were doing was then characterizing the proteins that are on the surface of these cells to get a better idea of what is required by COVID to enter versus what is required by regular SARS. So you can see, first things first, this is one of the first papers, there are some differences, right? So even though these viruses are related, they do not infect at the same level, and certain kinds of cells, these are all different kinds of cells. These are cell lines derived from different kinds of even animals, right? So up here they listed, these are all human cell lines. These are, you know, either monkey, pig, cow, dog, bat, you know, mouse, or hamster cell lines, right? And you can see in the human cell lines, if we just looked at those, right, you see similar patterns, although you do see some differences. For example, these CACO2 cells, which are actually cells that are from the gut, SARS-2 seems to be way better at infecting those. And these CALU3 cells, again, it seems to be better at infecting. Now, why is that? It could be that that's because those cells are, you know, maybe have some differential expressed proteins on it that we don't know about. So there's still some discovery to be had there. Or it could just be that this new version of SARS has evolved to be much more efficient in getting into the cells, right? What's interesting here, I want to show you guys something. This is a very interesting thing. You see this cell line right here? This cell line called MRC5 seems to be naturally resistant to both SARS and SARS-2. So what would you do as a scientist then to understand what's happening? What do you think? Why do you think they could be naturally resistant?
You look where the cell line comes from.
Right. So you look at where the cell line comes from, but what do we want to know about the cell line?
Its environment, or like its characteristics and how its membrane is composed.
Right. So what if we did, so one idea, this is one idea we could try, and it just, you know, came to me as we were talking, is look at a cell line that is easily infected by SARS, right? Like CALO3 or CACO2, right? And then do an analysis of all the genes that are expressed in that cell line, right? And then look at a cell line that is not easily expressed, right? Not easily infected by SARS, and then do a gene expression analysis of all the genes exposed in those cells. And what would you expect to see if you did that?
Common comparisons?
What would you expect to see?
Like
What might you find if you did that comparison? If I looked at all the genes that were expressed by a cell line that could be easily infectable by both SARS and SARS-2, and then I also looked at all the genes by a cell line that wasn't infectable by SARS or SARS-2, what would you expect to see?
Similarities?
different genes being expressed.
Differential expression of some gene, and that gene might be important for what?
For the infection of SARS-2.
Exactly. So that could be now a target or a therapy. So that might be a good experiment to try. Right? That's called a subtractive analysis, right?
And then,
So you're looking at the genetic sequence of all the genes being expressed by cells that are very susceptible to the virus, and then looking at the genetic sequence of all of the of cells that aren't infected. I actually, one of the best papers that I ever published in my graduate school did that for HIV. So we actually were able to find a protein that was made by a monkey species that naturally blocked HIV infection. It actually made the monkeys completely resistant to HIV. It was a genetic adaptation that they developed to make them resistant. And it's kind of amazing. And so researchers have still been trying to figure out a way to give that kind of adaptation to humans, but so far have not yet been successful. But with the technology being what it is today, let's say we found this gene or genes that made these cells resistant, we could put them in a lentiviral vector and then put them in the lung epithelium cells of patients with COVID. That sounds like a pretty interesting idea.
would you
What do you
do
think?
what we did with like SPOP with overexpression and underexpression?
Exactly. So if you found some genes that were underexpressed in these cells, we could try an experiment to see that if we made a lentiviral vector with those genes to make them overexpressed, then we would make the cells more susceptible to SARS or more resistant. Could we make this cell look like this cell by overexpressing genes that were underexpressed in this cell line? Make sense? Or are you confused?
Makes sense.
Makes sense.
It's actually kind
So
of
let's.
fascinating.
So I want you guys to team. How many folks we got on here right now? Even on this call, we're gonna do a quick experiment. We're gonna do a thought experiment. Okay. And hang on. Let me stop sharing my screen for a second. Okay. How many folks we got on? We got Alex, Gideon, Josh, Ahmad, Jalen. So I'm gonna say Alex and Josh, right? And then Gideon, Ahmad, and Jalen. I'm gonna split you guys up into two teams. Okay. One team, I want you to look up CALU-3 cells. Okay. And then the other team, I want you to look up MRC-5 cells. I want you to Google search those.
MRC-5.
Mm-hmm.
Okay.
And I'm going to do both on my end as well, okay?
CALU is a human lung cancer cell line.
So it's a human lung cancer cell line, right?
Yeah, the epithelial, like you mentioned before.
Okay. So
Commonly
human
used
lung
in,
epithelial cells, right?
commonly used in cancer research and drug development.
Okay. Talk to me about MRC-5.
Human diploid cell line used in production of viral vaccines.
Okay. So
Yes.
let's get more information. Share your screen. Share your screen on the MRC-5.
Okay. You
So
can.
can you see it?
Oh, it's mirrored.
Oh, okay, I see. All right, let's try it again. Let me try it on mine. I'm gonna Google search that. Let me see if I can find it. Hold on. MRC-5. Okay, let's see. Yeah, we can do the same exact experiment. So I'm gonna send you guys that paper that is now almost 18 years old that I did a long time ago. This looks like a human liver cell line. Okay. Let me see.
Yeah.
Actually, we're gonna look on the ATC site. Okay. So there's a place called there's a website, a company called American Type Culture Collection, ATCC. They're actually based here in Virginia, not too far from George Mason. I'm gonna share my screen, and they make pretty much all of the cell lines that you can buy commercially. All of these are cell lines you can buy, right? So if I look up this cell line, MRC-5, it tells me that it's a lung fibroblast from human cell lines, right? And it should give me some more information about it if I click on it. You could actually buy it. So I could buy these cells for $404. I could get a little tube of these cells, right? And then I could grow them up in tissue culture, right? And then I can take all of the RNA out of them, okay? And then I can sequence all of the RNA, all of the genes that are expressed. Okay? So we could do that. So you could get that information. So let's see, characteristics. What does it say about it? Okay, and this tells you, okay, so every cell line, okay, every time you have a cell line that people use in research, like these papers, the science papers that I'm talking about, they are generally derived from some sort of person or animal. So in this case, the MRC-5 cell line, can you guys see my screen?
Yeah.
The MRC-5 cell line was derived from a normal lung tissue of a 14-week-old fetus, right? In September of 1966. Okay? And it shows that it can grow a lot of different viruses. It can grow poliovirus, herpes simplex virus, vesicular stomatitis virus, that VSV I told you about, Okay. So what could be very interesting about this cell line is this person, right, could have had something in his DNA that made him naturally resistant to COVID. And that gene could be why some people don't get badly infected with it. This could be the whole, reason why some people do well and some people don't. So if we make the hypothesis that these cells somehow have some gene or set of genes that make them naturally resistant to COVID, maybe there's a population of people walking around with those genes. And then the other population of people that don't have them that are doing really bad. That would be a really cool study to look at. Don't you think?
Yeah.
Okay, so let's look up the other cell line. What was the other cell line called? CALU3?
Mm-hmm.
CLU-3.
It doesn't look like they have it. Let me go back and look. Oh, no, they have it. Here we go. CALU3. Okay. Let's look up information about that. But that I think that's a cool experiment that we should think about how we could do it. It seems pretty straightforward. We would just need to order the cells, and then we would take total RNA out of those cells, and then we would do some next generation sequencing on it. That's what we will do. And then compare the two subsets. Okay. Let's see if we can do that. All right, let's look up these cells. See, they cost about the same. This company makes a lot of money every year because all the labs in the world pretty much, will order from this thing. So if they're selling a little tube of cells for $400 each, they're making like billions of dollars. That's amazing. Okay, so this is a lung adenocarcinoma, so it means it was a tumor derived from a metastatic site. That means a tumor site that had some fluid around it, pleural effusion. Okay, so let's look at the characteristics. Okay, this was taken from a 25-year-old Caucasian male. The patient had received some prior therapy with some chemotherapeutics, it looks like, Cytoxan, bleomycin, and adriamycin. And this is a cancer cell. So the other one wasn't a cancer cell, but this cell is highly infectable with COVID, right? So maybe it's something about this 25-year-old Caucasian male genetically, right, that is different from that 14-year-old Caucasian male genetically that made one more susceptible and one resistant to COVID. Very interesting. So our mission as a research team will be to find what are the genes that are different between these two cell lines, right? Mm-hmm. And then if we found that, take the genes that seem like they made a person resistant and give them to a person who weren't resistant. That's one potential therapy.
How will we know the body will accept it? Do we know that other person is like how will we know that person's not susceptible to that?
What do you mean, Gideon?
Meaning that we know that this person is we will say like they're immune to that gene, but then we try to give it to the other person and they're not. How do we know their body will accept it or not?
You wouldn't know. You'd have to try it. And in the case of people who are I would estimate though that the potential for what you would call a autoimmune or not a graft versus host type of immune response would be kind of low, because I'm anticipating, unless, here's the question, unless the genes that were different between the two were on the surface of the cell. If they were on the surface of the cell, then there is a probability that you could get an immune reaction. But I think what you would then do is you would just take that patient. You could take DNA from that patient or RNA from that patient and make the therapy very specific for that patient. Because you would just take the gene whatever it is, the sequence of that gene in that patient, put that in a lentiviral vector that you then use to treat them. Right?
Mm-hmm.
That would be one way to get around that. Or, you could do a trial. You'd have to do some sort of trial to see what was the actual immunological danger of it.
Is there a possibility that like when scientists are evaluating this, they can miss something?
What do you mean?
Let me see how. No, you can come back to me. How I'm phrasing my question is not really.
That's okay. All right.
Like,
Go ahead. What were you saying?
like we evaluate all the sets of the genes, but yet we forgot this one thing. Never mind. You can come back to me.
Okay. Take your time. Formulate your question. Any other thoughts, guys, on this?
Would this be done under like a biohazard level? Like, I'd imagine we wouldn't be able to do this the same way we did with like the SBOP. It would be the same technique, but like there would be different precautions, right?
Yeah, you could actually do this same experiment, right? So I would take this experiment they did and just change it up a bit, okay? So here's what I would do, Alex, right? I would take all of the RNA out of these CALU cells, right? And maybe also take all the RNA out of these CALU2 cells. Then I would take all the RNA out of these MRC cells, and I would sequence all the genes in each one of those cell types.
And
then I would ask, what's the difference between at each gene, what's the different level of expression for each one of those cell types? So I would expect to see the same levels of these and these two that are equally infectable, but I'd expect to see a few genes different for this guy, right? And whatever those genes were, I would then make lentiviral vectors that express those genes, right? And then I would, either I'd have to see first thing you have to determine is what direction the gene expression change in. Did it go up or go down? Like I expect to see a change, right? I expect there'll be either some genes that are down regulated or up regulated in these cells that cause them to be less infectable by SARS. And this seems to work on all SARS viruses probably, right? All coronaviruses. This could be, these people could be generally resistant to coronavirus, right?
Are you pointing out the research paper? Because right now all we see is the shopping CALU3 thing.
Yeah.
Oh, sorry. Hang on. Let me stop share. Hold up. I was pointing at the research paper. Sorry. Let me share it again. Oh, it was sharing just my website. Sorry. Here you go. So look, here's the research paper. I was printing. So let's go back to what I was saying. So if I look at these cells, the CALU cells, right? And if I look at the Keiko cells, which is another cell that can be infected by both SARS-2, highly infected, and by both regular SARS, okay? And I took all of the RNA out of these cells, right? And took all the RNA out of these cells. And I also took all the RNA out of these cells and sequenced them. So I sequenced all the genes of all those cells. And I asked a basic question, which genes were different between, let's say, this cell and these cells, and this cell and these cells. And if I saw some sets of genes that were either down-regulated or up-regulated, then those are good potential culprits as to the genes that are most important for making these cells resistant to SARS. You see?
Yeah.
And so if I found what those genes were, I could then try to make a lentiviral vector expressing those genes or causing those genes to be underexpressed by using, let's say, a siRNA approach to downregulate, and then put them on these cells and then see if I blocked the infection.
Okay.
See what I'm saying?
Yeah, I see it now.
And then if you did, then this is now a potential therapy. That lentiviral vector, you could probably give it as an aerosol for people who had like really bad SARS, bad COVID.
So like it wouldn't be very biohazardous per se because the virus isn't experimented with directly.
That's right. Yeah, you're not experimenting just with the virus. You're making a way to engineer cells to be resistant to the virus. I'm trying to make these cells look like these cells, just like we're trying to make the tumor cells look like non-tumor cells. Same basic principle.
Right.
So one other idea is to see if anybody has already sequenced Calu-3 cells, right? So can somebody do a Google search for Calu-3 cell and gene expression, and for MRC5 cells and gene expression? Just Google search those terms.
Gene set, Calu-3, Mayan laboratory.
You share your screen now. Can you share your screen?
Sure. Can you see it?
Mm-hmm.
Right there.
Yeah, let's see. What you got? Yeah, so they have a gene expression cell line. Okay, this is very interesting. Let's see if we can download this. Okay, go back up to the top. Scroll back up. Harmonizome. What the heck is harmonizome? Let's see what this thing. So this is a cell line. 352 genes with higher low expression relative to other cell lines from GSC. So which other cell lines did they do it to? I want to know. See, so they found these different genes that were differentially expressed compared to other cell lines, but we need to know if the comparison cell lines were MRC5 or not, contain that. Let's just look at these genes though. Let's look at this list of genes. So this is a list of 352 genes.
And there's multiple pages.
Yeah. Okay, this is cool. That's a cool research. See if you can find the same for MRC5. Like bookmark that.
I can just come back to it.
Mm-hmm.
RC5. What was it called again?
MRC5 cells and gene expression.
There's not one for the same lab. This is a PDF.
You don't really want that.
No, this isn't it.
Go down, residual components, blah, blah, blah.
No.
I
wonder why there's one.
Go back to that website that you were on for the Calu-3 and see if you can type in
laboratory.
Mayan laboratory, whatever that is. See if you can search for MRC5. Or just MRC5. Look up MRC5. Nope. MRC5, yeah, put up MRC5 cells and see what you get. My point is, I'm trying to see if someone's already done it, right? It could have been somebody, these are common cell lines. So just like this harmonizome place or whatever had already sequenced it, we could look at gene expression.
No, I don't think this is.
That's not it. Okay, we'll go back. Go back a couple of pages to.
Calu?
Yeah. So this is interesting. I mean, one thing we would let's do a little literature search, everybody, and it's for the next session.
I think I found it. MRC5 cell gene set.
Yeah, where is that?
It's on the website. Do you want me to screen?
Show your screen.
Yeah, show your screen.
Yeah.
The grant access.
Yeah. Really neat. Can you do it or not?
Yeah, I just have to allow.
Oh, you have to go to your settings.
Yeah. I have to leave the meeting real quick.
Okay. So let me look that up. So anyway, yeah, let's see if anyone's done that research. So go back to your screen, Jalen, because this is what you would do as a researcher. When you have an idea, you're doing, first thing you see is, rather than you have to reinvent the wheel, has somebody already done it? Okay.
I put the link in the chat so you can share.
Oh, click on chat. Okay, click on the link in the chat, Jalen. See that chat from Alex? Click on that.
I have to exit my screen really quick.
If you go to chat.
I got it.
Share your screen again.
Paste. There you go. Thank you, Alex.
Okay. Let's see. There we go. MRC5 cell. So.
Oh, it's a capital M.
Yeah. So let's see what we can do with this information. 186 co-occurring proteins with the. So let's see, what does this mean? All right. So look, all of these they had different values to it. Scroll down. So it's a whole different set of genes. So one thing I want to do is, from this paper, from this something, download, like, make a spreadsheet of all of these genes for MRC5, and then also make a spreadsheet for all of the genes for CALU2. And then we're just going to see which ones are the same and which ones are different.
Ooh. So notebook? Notepad,
yeah, just make a, you got here. Who wants to do it?
Yes, not me.
Alex, can you do it with a spreadsheet?
A spreadsheet? No.
I don't know
how to
how
get the
to
data
do it.
from the website to the spreadsheet.
Yeah. Do you want me
All right,
to
let's
download
try
it?
it. Let's stop screen share. Send me both of those links in the chat.
Okay.
So one for MRC and one for CALU2. And I'll share my screen. Let me see if I can pull up a spreadsheet.
Oh, there it is.
Okay. All right, let's try it now.
There you go.
All right. Okay, can you guys I'm gonna share my screen? All right, can you guys see my spreadsheet?
Oh, you meant there? Yeah, I'd have to go to my school account.
That's okay. I'll do it from here. But what I'm gonna try to do from the Zoom. Oops. From not here. Let me see. I'm gonna get back to here. Oops. Okay, you're seeing my whole screen. All right, so let me see.
Do you mind if I go get cereal?
Yeah, go get some cereal.
Okay.
We're doing a little thought experiment here. I'm gonna have to stop my share for a sec though, and get back to the chat piece, because these are the websites. All right. Okay, so I got MRC5 and I got CALU3. Okay, share my screen again. Okay, so what I'm gonna try to do is I'm gonna try to select all these genes and see if there's a way to see all. There's 352 genes, and these are all differentially expressed. So let me see if I can do this. I'm gonna see if I can cut and paste direct. Boom, I can. Beautiful. All right.
Wow.
All right. Let me take this and I'm gonna move it down a bit. And I'm gonna call this CALU3. Okay. And then what I'm gonna do is do like each page, right? I'm gonna get all of them. Oops. Accidentally clicked on one. Sorry. Go back. Back to page 2. I'm just going to select. I can take those fields out later. 3. So we're going to make this is what's called making a gene list, okay? We're making a gene list, and then we're just going to compare in Excel. I'm going to make one for this. Oops.
Dr. Cunningham,
Yep.
what are standard values?
These standardized values are actually a change in gene expression. So this will be like, this is 1.8 fold versus normalized, right? So these, I think, are normalized gene levels. So what they'll do is they'll take, the way the sequencing works is it takes each gene and it tags it, right? And it tags it with a fluorescent tag. And that fluorescent tag will give off a signal. So it'll get a signal for each gene, okay? They have like libraries of these tags specific for each of about 25,000 genes that have been identified, right? Mm-hmm. Then they will normalize. They'll take a gene that's like in every cell, like actin, right? That it makes up the skeleton of your cells. You have pretty much the same amount of actin in most cells, right? And so they'll subtract that actin from the gene expression level of these other genes and then give you a standardized expression value, right? Mm-hmm. And that's what that is. And then you can actually start to calculate things like what's called log fold change. That means like how much is the gene expression different in one group versus another. And that's what I would actually do if I were doing this experiment just from scratch in my lab, right? I would literally take the RNA out of these cells, take the RNA out of those cells, sequence them both, and then compare the results. Okay? Comparative gene expression or subtractive. That's what I call it. So I'm literally just like looking at what's different between one group versus another. Right? So we got most of these guys. I think we're only 2 more pages of these. So this is for my CALU 2, and I'm going to do it for the thing. And then I'm going to see what genes are different. Like, and I'm going to send you guys a spreadsheet, and you guys can help me find what the differences are, because we're doing some detective work just with the existing data, right? And we might be able to find a potential culprit, right? In terms of genes that are different that make one cell COVID high and one cell COVID low, okay? So I'm going to call this CALU 3, but I'm also going to make a note that this is COVID-19 high infection, okay? Okay? So I'm going to call this the sheet CALU 3. Uh-oh, computer jamming up. No. Okay, and I'll make another sheet, right? And this is going to be my other guy. So this was my CALU 3. I'm going to minimize him. And I'm going to go to my MRC, okay? How many pages do I have of this guy? Ooh, I got a lot on this guy. Let's see. 186 proteins.
You can show more entries at the same time where it says show blank entries.
What do you mean?
Right where it says under genes. And then you can copy 100 at a time.
Thank you, Alex. Thanks for the common sense. Appreciate that. That's way better. Yeah, these are all genes, and they all have different functions, right? So what I'm going to be looking for when I do this is actually cell surface genes, right? Okay. I'm going to be looking for cell surface genes that could be different between the two, or Actually, anything. I want to see what genes are really different. They're most different between the two. Okay. All right. I'm going to email you guys this spreadsheet, okay? And clean it up and we'll look for it. Now, some of the X factors with dealing with someone else's data is you don't know exactly how they did it, right?
Mhm.
So I'd like to try to figure that out, okay? So we're going to look for genes that are different between here. So I'm going to call this COVID high versus COVID low expressing cells, okay? Okay? Okay. All right. I'm going to stop share. So I'm going to email this to you guys, right? And then what I want you to do for our next meeting is look through that list and see if you can find some that are different between the two. All right?
You want us to go through all the list?
Yeah. Go
Is this
through.
going to be on our Future Kings email, like the Google Classroom, or?
Yeah, I guess I can put it to Google Classroom. Let me figure out how to best do that. Or I can send it to Dr. King. He can send it out to all of you guys. Okay? But
Yeah.
I'm gonna email this to all of you, and then we're gonna do some research. And what I want you guys to do is, so in the spreadsheet, I think you can click on the gene, right? So let me share my screen again. If you find genes, you can actually click on them, right? Can you see my screen?
Mm-hmm.
So let's say we want to look at this NFU1. You can click on it, and it'll take you there. And then it has a bunch of stuff about it. Okay, it tells you about the gene. This gene encodes a protein that is localized to mitochondria and plays a critical role in iron sulfur cluster biogenesis. What I really want to see is genes that are what we call differentially expressed. So if this value is 1.4 here, okay, and the lowest value that this one goes down to is 0.2, I want to see, you know, A, what's different and how different is it? See?
You also want us to focus on why is it different, or no?
Yeah, we can start asking that question. Why is it different? But what I want to do what I want you to do is, for the genes that you find that are different, here's what I want. I'm going to add some things. Cell surface. Is it on the cell surface? Okay. So ask me that. Is it in the cell nucleus? Cell cytoplasm, rather. Okay. Or cell nucleus. So you have to look up information about the gene. And I'm going to show you guys a website. I'm going to include a link for a website called Gene Cards, where you can get information about every gene, right? So let me just put that there, okay? I'm going to show you this. It's a handy tool. We're just doing some detective work, right? It's for fun. That's how we do it. Cell nucleus, and then I'm going to say, is it an enzyme? Okay? That's what we want to know, okay? For each one that we find that's different. You don't have to do it for every gene. Okay, I'm going to put a link on here for Gene Cards. This is the site that I like to use the best, because this has like every gene and like everything about every gene. Okay,
Yeah, I'm back.
you got it? Did you get you some cereal?
I got some. So I kind of got tired of cereal. I mean, that's what I've been eating for like the past 2 or 3 weeks. So I made waffles and apples and peanut butter instead.
All right. So I'm putting this, gene information website below called gene cards, right? So
Mhm.
whatever gene you want to look at, let's say it's this one, Cas3, right? You can put it in gene cards and get the information about it. And it tells you all the different names for it. It tells you what it's known to do, right? And then also, if you keep scrolling down, it tells you every bit of information they have about this gene, where it is on the chromosome, everything. And then even if you scroll down further, this is what I love about this website. I love this website. It tells you where it is. So it's mostly in the
Oh, wow.
cytoplasm, nucleus, whatever. So it's like based on the research that's been done, it's pretty much in the nucleus or in the cytoplasm. It's not on the surface, right? I love this website because it has so much information. If I wanted to order a lentiviral vector that expressed it, I could do that here. They have different ways to actually do it. If I wanted to order an siRNA product that did this, they have it here, right? So you could order it. So we wouldn't even have to make it. So once we find the genes, we could change their expression one way or another without even making it. But the detective work is finding the genes that are different. And I want you guys, this is just one potential way to do this. I want you to do a little bit of detective work on your side to see what research papers have been published having to do with gene expression of CALU3 or MRC cells, okay? Do some literature search. So I'm gonna show you guys really quickly how you can do some literature search, and then we'll stop for the day, okay? So
Okay.
another website that we use in the lab is called Pubmed.
PubMed.
Yeah, I'm gonna put these links in this spreadsheet for you guys, okay?
Mhm.
Why did I go Gmail? That was force of habit.
One of the sites was Gene Cards, was
Yeah.
it?
Yep, gene cards. I put the site in the spreadsheet, but if you Google gene cards, it'll come up. All right, let's go. Yeah. Love that website. It's a wonderful website that somebody made. All right, let's go. PubMed. All right. PubMed is a public library. It's called the National Library of Science, NCBI, NLM, National Library of Medicine. And this is NIH maintains this. It's a repository of all of the published research papers dating back to, I think, 1930. Okay? I'm going to put that link here too.
Wow.
Okay? So you can go and search by topic here. So I can say CALU3 cells and gene expression and see what comes up. And it gives me 154 matches. But let me. This is a technique. I'm gonna see what happens. If I do that, it might not give me anything. Nope. And
I've noticed a
yeah,
common pattern. The search engines for most of the websites aren't that good.
huh?
The search engines for the websites aren't all that good.
No, they kind of stink, but you just got to do it. Oh, this is interesting. Look at this paper. Here's a paper that just came out in December. Competing endogenous RNA network profiling reveals novel host dependency factors required for MERS coronavirus propagation. Look at that. That sounds interesting.
They really do it all.
Hmm?
They do it all.
Yeah. So they looked at genes and that are important for MERS or not. And so we might be able to get some. Now, the one thing that kind of is not fun about this, and this is something that I hope will change in science, is that a lot of these papers, you have to have, they won't let you download them without buying them. But because I'm part of a university, I can generally get them for free. But sometimes you can't even you can't download the papers to look at them, which kind of stinks because I feel like all papers, particularly for coronavirus, need to just be free right now because everybody needs to put their brain
Yeah.
right. Okay,
Mhm.
I'm just downloading this, and now we can read through these papers. So I want you guys to look for some papers where they've done compared gene expression that includes either coronavirus or CALU3 or CACO2 or MRC5 cells. So I'm going to send you guys the cell paper that we were referencing, and then I want you to just do some detective work for the next time. And so our goal for next time is one idea that looks like we're gonna go down the path is see if we can find some genes that make certain cells more infected by COVID and certain cells less infected by COVID. Okay?
Mhm.
Based on the research that's been published. Okay, and then once we find those genes, we're gonna see if we can make lentiviral vectors that can make those genes. Okay? And then we'll do a basic experiment to see if we could change the level of gene expression of those genes in cells. And then we're going to find somebody who's doing coronavirus in the lab and send it to them. Right? Okay? Because we can't do coronavirus in my lab yet. They can only do it in a BSL-3 lab, but we could send it to them and ask to collaborate with them on a research project. Doesn't that sound cool?
Yeah.
Okay, cool. All right, let me stop my screen share. All right,
The U.S.
that's what we'll
has
do for
passed
next time.
Italy.
So I'm going to email you guys this spreadsheet and this original paper from Cell. I want you to read the paper from Cell and tell me what you do or do not understand. It's kind of heavy reading, right? But if you have questions about it, I want you to give me the questions. That's a real research paper. That's what scientists like me read. And I'm treating you guys like graduate students, so I'm allowing you to read, real papers. Because you guys are being trained to be real researchers. That's what we do. We read papers, we learn, that's how we get ideas. Okay? Any other questions? Thoughts? All right, well, good stuff, guys. Well, look, hang in there. Keep doing good. And, keep up your social distancing. What's up, Gideon?
I have a question. So, what happens if you know how the influenza comes like every season?
Mm-hmm.
Can't the coronavirus potentially come seasonal too?
Absolutely. That's actually probably what's going to happen. You're going to
So,
have seasonal waves of this.
yeah, because I heard there was another wave of the coronavirus happening in China right now.
Yeah. And they think it's coming in from people who are coming into China, right? And like resetting up the infections.
So, a funny question is, how long is online school going to be?
I think you're probably going to have online school at least through the fall.
You're kidding, right? You gotta be kidding.
I think they're gonna stop at the normal stopping point of the school year of June, but it'll be interesting if they even let people back to class in the fall. I think they will, but it's just gonna be different. Your class is not gonna be the same. You will not be as close to each other in the classroom. Everybody's probably gonna have to wear some sort of mask for a while.
If they provide some.
Yeah.
Or put that on our school supply list.
School supplies.
Everybody's just gonna have to wear a mask for a while. Everybody's no more handshaking. So handshaking is out.
Thank God.
What'd you say, Ahmad?
I think as long as we're safe, that's all that matters.
Yeah, that's the important thing. I mean, this is just to give you guys some perspective. This is so new, and it's only happened in the last 6 months. It seems like forever, but this virus didn't exist until 6 months ago.
It seems quick, to be honest.
I felt like there was early signs, but we just ignored it.
Yeah.
We did
Yeah.
for it.
Yeah,
Like the
this
way
country,
this happened.
the
In
United
the
States.
beginning,
Yeah.
we brushed it off. We are like, it's not gonna be anything serious.
Yeah,
But
I
here
think
we are
that's the
now.
biggest thing.
That was a big mistake, I think, by the current president and the administration that they didn't take it seriously. And
Procrastination.
they procrastinated. They wasted a good month, maybe a month and a half. And that has real consequences. I think there are a lot of people who unfortunately are going to die because of that. I mean, that's just,
Sad.
it's a sad situation. So this is why things like when you guys get older, they have a saying that elections have consequences. This is true. You need to go ahead and make sure you vote and make sure you got people who are there who can take care of the basic principle of keeping the citizens safe. So I'm praying for the current people. I hope that they can do a better job, but they did waste time at the beginning. I think, history is going to show that, and that's unfortunate.
But here's another question. So let's say we do find a vaccine for coronavirus, right?
Mm-hmm.
Now this is where we get into politics. A lot of people, there's going to be some people who are against vaccines.
Oh yeah, that's true.
What do we do?
I
What do we do?
forgot.
I mean,
Well,
you can't force. I mean, you can force people to do it. Just like for school, you guys have vaccines that you have to take in order to be registered for school. This will be one of those. It will be a requirement.
Okay.
So, I mean, look, it's still going to take a while. I want you guys to understand, like, it normally takes about 2 years to develop a vaccine, minimum, right? Because here's why. You don't know if it's going to work. And you honestly, with this kind of virus, which is called a respiratory virus, it could actually make it worse, right? Like, if depending on the kind of antibodies you generate, you could have some situation where the vaccine actually makes the infection worse. And so that's why they have to do the clinical testing. There's no shortcut. They have to test it in people to see, okay. And so think about how they're going to test this vaccine. It's a hard test because they're going to have to inoculate some people and then let them people walk out in the world and then see if they get coronavirus, right? So it's kind of like they're going to, and then so there's two ways they're going to test. They're going to vaccinate people, shoot them in the arm with the vaccine. Then they're going to take the antibodies out and see if it can block it in a test tube, okay? And if it can, right, then that's a good sign. But it still doesn't mean it's going to make the people have a better infection rate. They're going to have to track those people for at least 6 months to see if they then go to get infected. And they're going to look at the people who got the vaccine and the people who didn't get the vaccine and see what's the relative rate of new infection among them. And that's how you really know if it's working.
So all this is about timing. So this is going to be it's going to be a while until everything goes back to normal.
Yeah.
Right. And so that's what I'm saying. Even in the best case scenario, you won't really know if you have a vaccine until about 1.5 years, because you got to give it that time. You got to, they're already starting to vaccinate people. They're doing that. But then they got to wait like 6 months, maybe 9 months to see if those people then go on to get sick. And then they got to have a high enough number of those people that they've tracked to see, okay, out of these 100 people that we've vaccinated, okay, the people that we vaccinated 100 people, we didn't vaccinate another 100 people, okay? Out of the group of 100 that we vaccinated, only 20 went on to get the virus, right? Whereas the ones that we didn't vaccinate, 80, 90 went to get the virus. Now you know your vaccine is kind of effective. So that's what is happening. And hopefully by then they'll have some new medicines. They have at least 20 new drug trials that are happening. So they might have a drug that works by then. But even once you have that, that's why I want us to work on these ideas, because you're going to have new treatments, new therapies. The same thing might not work on everybody as well. So everybody has to put their intellectual capital on this and see if we can find some new ideas.
As I was looking at the gene sequencing, it said a lot of Caucasians, but this virus is like based off the news, it's saying that it's affecting more blacks.
Yeah,
Yeah.
more black people.
More minorities.
Yeah. And that's a complicated question because they don't think it's necessarily genetic, right? They think it's more that black and brown people have traditionally had poor health care. So if you've had like conditions like diabetes or hypertension or things like that, it can really make this virus much more dangerous.
But at the same time, I feel like they're getting their data based off people who are living in poor areas too.
Cities. So more black and brown people live in cities, and the outbreak is now worse in cities right now.
Mhm.
But they're also seeing in places where even aren't large cities, like in Michigan and other places, in rural Wisconsin, they're seeing more of a disparity. It's called a health disparity of African American and Latinos getting this, and particularly African Americans. It seems like in certain cities, like in Chicago, 70% of the coronavirus deaths are African American, and African Americans are only like 25% of Chicago, right? So it's a really big problem. But a lot of that has to do with access historically to healthcare and overall like health, right? You have heart disease, diabetes. If you have any of these conditions before you get coronavirus, it'll make the coronavirus much more deadly for you.
So like say there's a vaccine, right? Do you think even after the vaccine, it changes our belief on human interaction?
No, I think this will for the immediate near future, at least for the next few years, dramatically change how we interact. I think handshakes will probably disappear, right?
I think I'm gonna get tired of Zoom calls.
I think everybody's going to get more used to Zoom calls, and we'll have some different technology. I think we're going to develop some hologram type technology and
I
things
know,
like that.
it's so cool.
We could do. I think that's going to happen. And then I think people are just going to have to learn how to readjust to less contact. That's going to be hard psychologically for people because we're very social animals, right? We like to be around each other. And it's actually important for us to be near each other. So it will be tough, but we're going to just have to
5 months of this. I'm going to go crazy.
I know, right? So you just got to keep contact with your friends on Zoom and everything, and just try to connect and try to stay positive. I mean, you guys are lucky that now you're learning how to be scientists. You can be part of the solution. Just start reading, right? Read more about it, educate yourself more about it, and start trying to come up with ideas. We're a little research team here, so we're trying to figure out new ways to attack this thing. And so we got one idea that we're gonna track down, looking at the differential gene expression, and then I'll talk to you guys about another idea I had next time.
Okay.
All right.
Okay.
All right.
All right.
All right. We're gonna sign off, guys.
Thank you, Dr. Cunningham.
Yeah, thank you.
We'll talk to you.
See you next time.
See you.
Bye bye.